Good morning, and welcome to the fourth webinar of the 2022 Global Year for Translating Pain Knowledge to Practice. I'm Benjamin Farrell, Coordinator of Global Programs at IASP, and we are excited to present this webinar and hear from our speaker. Today, we will be hearing a presentation on translating genome-wide transcriptomics data for understanding the pathophysiology of human pain. IASP are thankful for everyone who could join us today and remind you that today's seminar will be recorded and available for later viewing at the IASP's Pain Education Resource Center. I'd also like to encourage our viewers to utilize Zoom's Q&A feature to ask any questions you may have during today's proceedings. During this webinar, I'd also ask for everyone to remain muted and cameras turned off to avoid any distractions. At this time, I'd like to introduce our moderator for today's webinar, Jeffrey Mogul. Dr. Mogul is the E.P. Taylor Professor of Pain Studies and the Canada Research Chair in the Genetics of Pain at McGill University in Montreal, Canada. He's also the Director of the North American Pain School, or NAPS, and has made groundbreaking contributions to the field of pain genetics and has written many reviews on the subject. With that, I'd like to thank you all again for joining, and I'm happy to pass today's proceedings over to Jeff and look forward to hearing from our speaker. Thank you, Jeff.
Thank you, Ben, and thank you everybody for joining us here today. It is my great pleasure and honor, that's with a U, by the way, to introduce our speaker today, my close colleague and friend, Dr. Luda Duychenko. Luda is the Canada Excellence Research Chair in Human Pain Genetics here at McGill. For those of you who don't know, this is one of the most prestigious academic positions in our country. Luda is part of the faculties of medicine and dentistry here at McGill. She received an MD and a PhD in molecular biology from the Russian State Medical University. Started her career in industry at Clonetech and Adagene, during which she developed some very widely used and widely cited molecular tools that are still in broad use today. She shifted over to academia in the year 2000 at the University of North Carolina, and a number of years ago we were lucky to poach her over to McGill, where she's been ever since. Luda is going to give a talk today that is at the very forefront of techniques and methodologies that really are all about translation, which of course is the purpose, the theme of the global year, a way to use humans to figure out mechanistic hypotheses about how chronic pain works, hypotheses that then can be tested in animal models and then brought back to humans again. And I think Luda is going to give us a number of examples of that sort of translation from one species to another back and forth, which is a very exciting development in pain research. And so I'm looking forward to the talk. I hope you are too. So with that, Luda, please take it away.
Thank you, Jeff. Thank you for introduction. So as we know, I will talk today about the translating of genome-wide transcriptomics data to understand molecular pathophysiology of human pain. And so I often ask myself what we know about pathophysiology of chronic pain, how we think about this. And so this slide from Aaron Tracy from review 2008, I don't think, our understanding changed much. We understand there is a central sensitization, which leads to some intermediate phenotype associated with chronic pain. We also know there is some peripheral activity which lead to central sensitization or initiate one. But molecular pathophysiology of chronic pain condition are largely unknown. In other words, we don't know what actually molecular events in what sequence happen in human body leading to chronic pain. We know much more about molecular setup for hypersensitivity. So in other words, we understand much better how we feel acute pain, right? But not chronic pain condition. So what I would like to do, what I would like to do with my next few years is to try to understand this human molecular pathophysiology using human data, human molecular data. So what are molecular data? Well, as we know from central dogma, we have DNA, which is transcribed to RNA, which translated to protein. So also protein actually working here and produce some small molecules like serotonin, for example, or lipids. And all this together is leading to manifestation of human diseases. And of course, we all here, we study pain. So, so the closer we are to phenotype, is it small molecules, protein, RNA, DNA, the better our statistical power. So we can understand, we need less people to associate it with disease, but there is different other prices to pay for it. So today I was contemplating about telling few stories, so tell one story, and I decided to actually tell one story, but well. And this story will be about translating full transcriptomics into understanding of molecular pathophysiology of acute to chronic transition. So this is a recent paper we published together with many colleagues and with Jeff also in Science Translational Medicine. So we started this journey. It actually was long, because now I will tell you the story. It's all looks quite obvious, at least obvious to me. But it took us a few years. And it started from this cohort, which was carefully, meticulously collected by Massimo Allegri in Italy. This was a acute back pain cohort where we had 100 Caucasian patients, males, females, at the acute stage, 6 months of the acute pain episode and 3 months late. So we have this T1, T2 time points. So in 3 months, some of the people resolve pain and some of the people didn't resolve pain. So they stayed with persistent pain. So we call them resolvers and persistent. So when we analyze this data, and so the first author of this paper, Marc Parisien, so he analyzed this result, left, right, up, down. So when we analyze this data, the most striking difference and most informative results we had is when we compare the transcriptomics of the same people over time, right? And compare, for example, resolvers, not resolvers at acute, because we want to identify markers, and we also look at the 3 months time between resolvers, non-resolvers, what's the difference. But believe me, the most informative way, and right now we look already on the many transcriptomics data set, is to actually look what's happened over time in the same people or in the same group of people. So this is volcano plot, and you see all these nice pink dots. This is, each dot is the gene, and this is all gene differentially expressed in resolvers between acute stage and pink, which is magenta. It's above genome-wide significance. So after correcting for all genes in the genome, this is how many genes was differentially expressed. in people with who resolve pain. You see the number of the genes, so altogether it's add up to almost 6,000 genes, so which is basically almost half of the all expressed genome in blood. And what's happened in people with persistent pain? Exactly nothing. So it's completely collapsed differential expression. So they kind of frozen in this stage. So we'll look if it all can be explained by the blood composition, and we look if there is a significant difference in the blood cells, which we reconstructed actually from transcriptomics. And again, we saw significant difference in the blood only in resolvers, but not in persistent people, such as the neutrophil number would go down in the people who resolve pain, mast cells go down, T cells go up, and NK cells go up. Absolutely nothing happened in people with persistent pain. And by the way, this one, if we control for the change in cell composition, we still will have basically, mostly the same number of genes. So the gene difference is not mostly because of the difference in blood composition, but it's mostly because of real differential gene expression. So then we did pathway analysis. So pathway analysis, when you combine all these genes, I mean, there's really too many genes. So it seems at that point pointless to try to figure out which gene exactly, right? When you have 1,000 genes, okay. It didn't seem productive to look at each gene. So we was looking for the way to combine this information, and we did pathway analysis. When we did pathway analysis actually had much more statistical power. So we had many pathways which were different, which were differentiating in the people with resolved pain. So then we decided, okay, so let's start from somewhere. Let's start from acute stage, and let's compare resolvers and non-resolvers, people who resolve pain and not at the acute stage. And let's see what pathway were the most differentially expressed. And if we go through the kind of non-specific pathways such as protein synthesis, right? Of course, it will be protein synthesis. If we will look for the kind of biologically meaningful specific pathway, we saw that there's inflammatory response was the most significant one. And this would be fine, would be expected, if not the direction, because direction was such as that people who resolve pain has much higher inflammatory response at acute stage. So you can see this bar, it means it's going, it's up in resolvers, near inflammatory response, acute inflammatory response, and all of them was going down very fast down in resolvers over time. So there is a high inflammation at the acute stage, but it's then go down, right? And again, in persistent, really nothing happening in people with persistent pain. So the next, or actually even more significant pathway, depends how you look at this, at time 0 was the leukocyte activation, basically driven by neutrophil activation and leukocyte degranulation. So it also was much higher at people with resolved pain, and this pathway also was going down over time. It's kind of was going down also in people with persistent pain, but at much smaller magnitude, because you see the y-axis here is logarithmic. So we also look at correlation, right? Especially because, here we have kind of the same pathway. So we said, like, interesting is the people who resolve pain and don't resolve pain, are they doing something opposite one to each other in terms of the pathway? And the answer was no, they actually doing pretty much the same thing, or at least the immune system doing pretty much the same thing. So here's the correlation of pathway between resolvers and non-resolvers. So, very significant statistically. R square 42% means that, it's very statistically people, immune system doing the same thing. But what is the difference? Well, the difference is the magnitude. So the golden line here, if they will have a perfect correlation, magenta light is the real correlation. So we can calculate the alpha here between magenta and pink, and this alpha is basically corresponds 30-fold difference. The same process kind of capture in this graph. You can see that it's a test statistic, means how differentially expressed gene, and you see in resolvers it's kind of distributed in terms of how different differentially expressed gene distributed both up and down regulated. In the non-resolvers in red, it's collapsed, so they don't really differentiate their genes. And again, here we can calculate how much, and it's 30-fold. Right? So it's not really in this cohort, in this time, we didn't see not really direction, it's a magnitude. So at this point, we was looking for a replication cohort, and we couldn't find the low back pain acute to chronic full transcriptomics, but we had TMD, facial pain, oral facial pain, musculoskeletal pain, and this was so-called opera cohort from UNC, directed by Bill Maxner. In this case, we also had people with acute TMD facial pain and then resolvers in 6 months. So first of all, you can see the cumulative fraction of differential expressions. Remember, I showed you that how those who don't resolve pain, so their differential expression is collapsed, right? They don't really regulate their genes very much. And those who resolve pain have much broader gene regulation. And in TMD, so yeah, we can replicate also the pathway. So basically you see the inflammatory response, leukocyte activation, leukocyte degranulation. In resolvers, it was quite significant. Was down-regulated over time and up-regulated at acute stage. And the same true for leukocyte activation and leukocyte degranulation. In opera, we also had controls, and we have a very chronic pain patients. So we can kind of look at the more broader spectrum of event. And so what maybe I can start is from the chronic, right? Okay, so here we always compare different stages, and we look for different this 3 major pathway we kind of was concentrating on, and we compare them with healthy volunteer. So statistically, neutrophil activation and degranulation was significant in chronic versus healthy, but in the way we kind of knew before, it such as the test statistic is positive, meaning they were activated in chronic pain patient. So even at the acute stage, yeah, we have a much higher inflammation and neutrophil activation for resolvers. But if you go, long enough, then it's chronic pain patient have this inflammatory response. But you can see even by test statistic, it's very mild, right? And we already know this. That's why all this result was counterintuitive for us, because we know that chronic pain patients, they have some type of low-grade inflammation. Okay, yeah, this is true, they do. But if we will look at the resolvers at time 0, for example, let's look at the neutrophil activation. So test statistic positive means they really have a very good inflammatory response, but at 3 months they have a negative, very significant test statistic, means they not it only go down, but they kind of undershoot the explanation, right? Inflammation. So they really resolve and they trigger anti-inflammatory response. So this is what we kind of reconstruct from our data. So people who resolve pain, okay, so the pain go up and then they go down, but the inflammation is going really up at acute stage and then resolve. When the persistent pain people, inflammation actually also start a little bit higher at before pain, and this is not from our data, but this from the literature more. So it's increase very little, but then it doesn't resolve, but it's continue into chronic pain. So at this moment, we actually had a lunch with Jeff, and I was sharing with him all our results and confusion I have all about these results. So Jeff was telling me, okay, so what you're telling me cannot be, because we know for decades that if we inhibit inflammatory response or any immune response for that matter, like with dexamethasone, it's analgesic, and so cannot be. And then Jeff said, well, how long we usually watch the mice after we treat them with the drug? We don't. So let's do this. I mean, it's you know, it's straightforward experiment. So let's do animal study. So we did. The Lucas Lima was the postdoc in Jeff lab who did then all following animal experiments. And actually, he also made the slides. So thank you, Lucas. So first, we did a very simple experiment. So again, let me say, let's say Lucas did all the simple experiments, and he started with CFA, with the complete fruit adjuvant. So just like inflammatory, good inflammatory pain in mice, and so he treated mice with dexamethasone for 6 days. So this is what we get. Originally, yes, CFA, threshold of 1 fry, day 3, day 6, the mice develop hyperalgesia, which then resolve basically in 2 weeks. The mice treated with dexamethasone, they have a very significant analgesia, but then it switched and they develop hyperalgesia. At that point, Jeff and Lucas, they share with me this data, say, look, day 35, it's still like a floor hyperalgesia. And it doesn't seem that it was related to edema, because, so first the edema was a little bit reduced by dexamethasone in the pore, but then it was really no different. So it's not because of the edema. And then, of course, we was waiting when mice will finally come to the baseline, and it took them 140 days instead of, you remember, the 12 days. So then Lucas also repeat this experiment with in CCI model, in neuropathic pain model, not the same magnitude. And yes, dexamethasone, as we know, don't produce analgesia in this model, but it's very significantly prolonged resolution. I mean, using dexamethasone at acute stage, very significantly prolonged hyperalgesia in this mice. The same true for NGF, this model which reviewer, thanks to reviewer, asked us to do this model of so more maybe closely to low back pain, and again we had the same thing. So it was actually, yeah, it wasn't statistically analgesic even in acute stage, but then it's prolonged hyperalgesia in mice. So we actually then tried to other model, and Lauren Martin in Toronto tried in postoperative pain in foot incision, and you see how robust, even more robust, the po-incision model for postoperative pain. Again, the mice who was treated just for 6 days with dexamethasone and acute stage have really prolonged hyperalgesia, lasted for up to 150 days. And by the way, I didn't mention, but you can see there is really no sex difference. So once sometimes it's happened, there is no sex difference. Both males and females show exactly the same things. So another things which also Lucas wanna try, he said, well, if it's a really inflammatory response driven by neutrophils, how about ice, right? So we use ice basically with the same intention to reduce inflammation, because it's good to reduce inflammation in acute stage. Okay, say if I model ice, you can see analgesic, not sure if it's statistical or not at acute stage, and then long hyperalgesia. So the next question was, well, is it really inflammatory response, which is important not to inhibit, or it's somehow pain which we should not inhibit? And so we compare different drugs, NSAID, gabapentinoid, sodium channel blocker, and opioids. So the same setup. So what we saw, it's only NSAIDs which produce this long-lasting hyperalgesia, not gabapentin, not lidocaine, not morphine. You can see diclofenac, NSAID, the green one, is that one which was different from everybody. You may see presentation here for duration is very different from everyone else. So, okay, then with our pathway analysis was saying who believe pathway analysis, not everybody, but yeah, maybe pathway analysis told us truth. It's really inflammatory response driven by neutrophils. Okay, what if we will remove neutrophils? So what Lucas did is remove neutrophil with antibodies for 20 days. And in this case, without dexamethasone, just removing neutrophil, we receive, yeah, basically a very similar picture that it's mice treated with neutrophils, they show higher threshold withdrawal, but then it switch and they become hyperalgesic for 120 days. And the last animal experiments I would like to share with you is when we say, then if it's really neutrophils, can we counteract dexamethasone if we will either give mice neutrophils or this protein S100A8 or S100A9, which is kind of the main protein which neutrophil produce and secrete, and they specifically secreted during this acute inflammatory response. So by the way, so we also we landed in these two proteins also when we look at the human data, because neither our who represent epidemiological expertise in our group, he said, well, okay, let's look at the molecule which will be the most differentially expressed between resolvers and non-resolvers in the acute stage, and then go down with the fastest speed in the resolvers. So Mark Parisian, you know, just look, it was like all in the same Zoom call, and he looked and he said, okay, S100 AA. And yes, look, guess what is the main protein in neutrophil. Okay, can we counteract dexamethasone? Yes, we can. So now the black line here is the dexamethasone you see in our model. If we treat mice with 6 days dexamethasone, they develop long-lasting hyperalgesia. But if we either introduce neutrophil or S100 protein to mice, the mice we completely restore their sensitivity to warfarin, so they completely not hyperalgesic anymore. So it's really neutrophils looks like. Okay. So what then became clear from these experiments, or at least this is the only interpretation we can offer, is that there is this acute immune component. There are acute immune processes that confer adaptation at acute pain stage, and impairment of such inflammatory responses might increase risk of chronic pain development. So if this phenomenon seems in human, it could cause dramatic implications for treatment of acute pain. And then we said, okay, can we have any evidence that this is taking place, this phenomenon? So what kind of human data we can take at this point? Of course, the best ever will be clinical trials, right? So have some type of post-operative pain, acute back pain, and look at the use or not use NSAID or dexamethasone or other type of steroids. Okay, so everybody who right now listening me, please think if you can do this clinical trial. But we didn't, we could not. So, but we could look at the UK Biobank. So in this UK Biobank is 500,000 people, which has been recruited, and with many phenotype that has been collected on these people, including pain phenotype. So specifically, we look at 2 questions. So first, we consider people who have acute pain if they answer question, in the last months, have you experienced any of the following interfere with your usual activity? And then the question was any pain in different part of the body. And the next question was, have you had this pain for more than 3 months? So in UK Biobank, there is 8 body sites. We look at the back pain. So the acute pain people said, yes, I have pain for last months, but it was not lasted for 3 months. And okay, so we took these people, and then UK Biobank also have 2 other enrollment wave, so 2 other visits between 2 and 6 years. And so we then see who of those people who reported acute back pain and enrollment also said 2 to 6 months later that they have a back pain, a chronic back pain. In other words, they would answer the question, yes, I have back pain, and yes, it's more than 3 months. So now we also have a drug usage on these people. So we ask question, if the drug usage affected the probability of them still reporting chronic back pain. Okay, so this result has been done by Vivek and Jonathan under the guidance of Audrey Grant in McGill, in our human pain genetic program. And what we saw is, so we had enough people to analyze the effect on pain chronification for NSAID, paracetamol, and antidepressant. You can see in the single model, the NSAID was very significant, 1.7 times more like people who take NSAID at the acute stage, 1.7 times more likely will still report chronic pain many years later, not paracetamol and not antidepressant. So model 4 is a full model. When we use all drug as a covariant, it still was significant. In fact, it didn't change. So we also use the psychological distress and number of painful sites as the proxy at the acute stage as a proxy for pain intensity. And again, didn't really change our results, the same 1.7-fold more. Likely people who take NSAID will still report chronic pain. And the last model, we look at the neutrophil percent. And so people point me that this looks like a 0, but it is actually statistically significant difference that kind of consistent also with our, it's very small effect, but it's significant and it's consistent with all our results. That people who have more neutrophils at the acute stage, they're less likely to still have chronic pain many years later. So, okay, I'm at the conclusion slides. So I have a couple of those. So what, to me was conceptual breakthroughs. So it's looking at all these results make me think differently about pathophysiology of chronic pain. Yeah, and we're talking about translation of our data, because remember we started from transcriptomics, right? Okay, so the first breakthrough for me was that we generally, I generally, and I think we as a field generally think about chronic pain is active pathological process, right? And so we need to inhibit this process, and once we will inhibit it, then we will prevail. But from our data, it looks like the pain is the absence of the active adaptive processes. So we need to study what should go right, instead of what went wrong. So we generally try to find the pain gene when, yeah, it's really no one single gene, right? There is a pain pathway. There is a huge pathways that underline pain resolution. So there are many of them. So we generally measure pain intensity in mice, and we ask people when maybe we should be more concerned about the pain duration. So finally, we generally think about pain resolution is somehow linearly developing process, but it can be conceptualized more like a spirally developed inverted U-shape. And why I said why U-shape is because so we have first this adaptive inflammatory response, and then it's go down, right? And if it's not going down, this is the problem. But it's also looks like we need this peak of inflammatory response potentially to trigger anti-inflammatory response. Without this, it just doesn't. Inflammation doesn't resolve very well. So why this word spirally develop is because we saw that, okay, neutrophil goes down, but T cells and NK cells go up. It's maybe also it was macrophages in between, which we just didn't capture because we only had 2 time. So I think it's a spiral of good event, it's right event which need to happen. But it's not linear. It's not like if you have good things that you have more and more good things over time, and if you have a bad things, it's a more and more bad things over time. Okay, and so on the level maybe of conclusion, let me share with you kind of a few of them. Looks like there is an active adaptive component of immune system in acute to chronic pain transition in low back patient, and likely other chronic pain conditions like we saw TMD, and like we saw in animal model, we saw in basically any animal model we try, we saw the same effect. So inhibiting this intermediate inhibiting this immediate, active, adaptable, I don't want to say adaptive in the immune sense, but adaptable immune responses in the human with physical injury may lead to pain chronification. So which means that in the drug development protecting against chronic pain, our animal model maybe should target pain resolution rather than just blocking pain, right? So maybe we should pay more attention to pain resolution rather than just developing drug that analgesic, because analgesic drug not necessarily affect what we discussed. It's not on the pathophysiological pathway necessarily. In drug development, protecting against chronic pain, we may target activation of immune response instead of inhibition. So we always think about inhibiting something, right? Like what our data said. So wait a minute. So maybe we should enhance. Maybe we should enhance our resources, and maybe what we know what is working. Physical therapy. What is working for chronic pain? Physical therapy, acupuncture, activated thrombocytes, activated plasma. Yeah, exercise, physical therapy, did I say so? But all of them will lead, all of this treatment will lead to activation of our immune responses. So maybe so we need to pay more attention and look how this treatment are working. And the last one, which probably last, is what occupied 90% of all press release of our paper, is that despite of high analgesic efficacy at early time point, the inhibition of acute inflammatory responses and pain management may be counterproductive for long-term outcome of chronic pain patients. Okay, well, I would like to thank you very much for everybody who come, and thank all the people, our program on the human pain genetics, and the funding from both US and Canada, who actually allow us to have time, and we have time to think about our results. We have money to do all these expensive molecular tests. And thank you again.
Well, thank you very much, Luda. That was great. While people are thinking of questions to ask, which are likely going to be about the story you just told, I wondered if I could start a little conversation with you to try to put this into some perspective and ask some bigger questions. So you showed a very successful translational story with transcriptomics, which of course is the study of the expression of genes, right? The number of mRNAs in a place. People watching might be more familiar because it's older with genomic techniques, which of course are the study of DNA variants. And I wonder if you could comment a little bit about the advantages and disadvantages of each approach and your thoughts as to the translatability from one species to another, say, of both these approaches.
Yes, will be pleasure. So first of all, so DNA. So when we talk about genetics, we usually think about DNA, and we usually think about SNPs. We usually think about differences in nucleotides. And yes, we had we have much more data from this domain, like GWAS. In this
genome-wide association studies, by the way.
Right. So in GWAS, when GWAS is started to be possible in like feasible money-wise, which was probably about 10 years ago, I would say, the people was very excited, and they said, okay, now we will know everything, because now we will find these few genes, and we will we will figure out how we need to treat pain and other diseases. And it was a lot of disappointment overall in the field of genetics, because people didn't see a huge effect of a few genes. And now when we're thinking back, we say, okay, of course it's the case, because all our experience was coming from the rare mutation, like sodium channel NAF 1.7. So there is a one gene, the gene completely destroyed particular family, some very severe phenotype appear, right? Like complete congenital sensitivity to pain. But this doesn't happen for common diseases. So like low back pain is a common disease. It doesn't happen there. So there will be very few people in the world, or limited anyway, which actually has mutation. But absolute majority, in majority of the people, it's genetic component fitted by very few common genetic variants, and it's completely depend on the environment. So it's a gene environment interaction. So now we have not one, not two, not even 100. We have thousands of genes contributing at a very, very little extent. Okay, now we still
can I stop you for 1 second and ask a
sure.
little follow-up? Okay, so you say there are thousands of genes. Is it possible to find them, and would it be worth the search to find them?
Well, there are a lot of people try to find them, including me and my colleague. So using all these genome-wide approaches, when you don't have the specific candidate genes, you look at the old genomics. So the disadvantage, and I was getting into the disadvantage, you need really hundreds of thousands of people to capture the effect. Yeah, this is the problem. But the advantage of genetics is it's never change. So once we bought with this, we carry it. So it doesn't matter when exactly you take DNA and when did you look at the SNPs. It will be always there. When with transcriptomics, the effect is huge, but and you need supposedly much less people to see the effect, but it's changing. We saw even, it's not even changing, it's expressed, not expressed. It's expressed high, very high, and then very low, and it's all happen. It may happen very quickly. So we need many, many repetitive samples to see what's happening.
Okay, so let me ask a follow-up, because, okay, so you need a lot of time points, and that remains a practical challenge. The other practical challenge, of course, and the other big difference between RNA-based techniques and DNA-based techniques is DNA, it doesn't matter where you take it from, it's all the same. RNA, it very much matters where you take it from. So I wonder, and of course in humans, really the only thing mostly you're going to be able to get your hand on is blood. And so I wonder if you could comment on what we know about gene expression in blood versus gene expression in the dorsal root ganglion, the spinal cord, the anterior cingulate, areas where if it were possible and ethically permissible, we would probably want to take our tissue.
Yeah, this is also a very good point, of course. Also, the DNA is good, that DNA is the same in any tissue. Do we believe that pain is in the brain, or we believe that pain is in the immune system? Well, this data says that at least pain can be initiated by immune system. And I think, it will be not a bold statement by now if I would say it's probably near immune interaction, a different stage of development. Then the DNA, even taken from the blood, carry the same SNPs. So then we can then do some techniques like partition heritability, when we take all this genetic variant and kind of map to where these genes express, and then try to reconstruct using this technique, and this will be tell us, exactly about the neuronal system, what's happened in neuron. Now, the blood, of course, has disadvantage because this is not what we think the pain is. And besides that, maybe it partially is, maybe it's very important. Again, our data says it's very important, the immune contribution in the initiation of chronic pain. Besides this, there is actually a lot of correlation between gene expression in the brain and gene expression in blood. In fact, the highest correlation between different tissues, if you look for the blood, because blood, of course, advantage of being accessible for tasting, it's brain. So the correlation between blood and brain is very high, much higher than between, for example, blood in the liver or anything else, basically, more or less. And with DRG is even more, because DRG is outside of blood-brain barrier. So blood cells, more or less, I mean, some people will now agree it's not exactly that, but more or less going back into, DRG back and forth as they please. So this is something also, we would like to study more. What's actually happening? Why inhibition of inflammatory processes lead to pain? So in the next organ, logically, we can look. It's DRG, or we should look probably. And again, now this is the power of the mice study. And so with mice, we can do stuff. We can isolate the DRG. We can inhibit a particular pathway. So we absolutely need mice research for mechanistic studies.
Thank you. Okay, let's go to a question from the audience. Here's one from Suleiman Dibhaj. I'll try to paraphrase this. So he's talking about the different types of gene variants that could affect the pain phenotype. And of course, there are ones that change the actual coding sequence, and then there are regulatory variants and silent mutations. I'm wondering if you could comment on what you think the contribution of the variants with obvious effect on a protein versus the more subtle variants. And so in 1,000 years, when we understand all this, what do you think the relative importance of these types of changes are going to be?
Well, it's a tricky question. It's not an easy question. Why? Is because on one hand, right, the variants which affect expression of the gene has the which affect gene existence, I would say. Like, so the stop codon, okay, in the beginning of the gene, you introduce stop codon, that's it, you don't have any more the protein. Of course, it's the most severe one and will have the most effect, rather than the most subtle difference when you have so-called non-synonymous changes. For example, change of leucine to tryptophan, for example, right? So probably more severe, versus you just have a slight difference in the expression of the gene. So you would think that the dose which is in protein, and especially which affect the mere existence of the protein, will be the most important. But it's not. Why not? Well, because they're very rare. So we come back to this problem of the rare familial mutation. Yes, they exist, but they don't define the frequency and development of disease in the general population. In general population, it's really this variant which mildly change the functional expression of the gene seems to contribute in the bulk. This is the one which contribute. We can look, and right now there is a lot of interest to it, including our lab, when we look at the exome sequencing, when we look at the rare mutation, and then we can see like real association with chronic pain with the effect size of 1,000-fold. In other words, you have this mutation, definitely have a gene, but it's very, very rare. It's affect again few people. we can learn about pathophysiology this way, because we can say, okay, this gene is important for pain, yeah, but it will not explain us the dynamic, I would say, of development of chronic pain.
Great. Let's move to another question from the audience. This is from Abdulaziz Hakim. What in your mind would be the next steps to understand the precise mechanism that either resolves pain or, in contrast, lets it transition to chronic pain? Right. What are you looking at as the critical set of experiments to be done there? And are they mouse experiments? Are they human experiments? How do we go further here?
Yeah, right. I would like to know, Jeff, your opinion too, but let me start. Okay.
We'll save that for my webinar.
Well, I think we should definitely do both, and we can learn different things from human and mice, and even each mouse experiment will have restriction and limitation and advantages, and the same each human experiments. So in a human, what we're doing right now is we collecting human samples with much higher frequency. So in other words, we would like to see what's happened over time with different pathway and how one pathway kind of start the other. The problem, of course, is what we saw is we need to look for the black cat, which is not even in the room. So we need to look what is absent in people who develop chronic pain. So we need to see what's happening in the people who resolve pain, and then try to understand which of those processes we observe relate to pain, to pain actually, right? So, and this is what mice will come handy. This is when we will come back to you again. I'll say, okay, we have this, and this, but we don't know what is related to pain and what is don't relate. What is related to pain resolution? So then we will start to block, I guess, one pathway after another in a different time point. And from this perspective, we also right now in my lab, we take mice and we take very, very frequent samples of the mice, and also again, thanks to mice contributing to science, blood, brain, spinal cord, DRG, and see how all this resolution pathway develops in different tissues.
There's one question here. Do you think any of this represents or could soon represent a biomarker of pain? This is a question from Maria Tarosa.
A biomarker. We all want a biomarker. Me too, and especially because this is what NIH give money for. From what we saw, the biomarker, maybe we should look at biomarker not kind of at the acute stage, but the speed of change. So trajectory. I think this should be this is the direction at least I'm looking at, and I'm thinking, well, in other field we have this example. In the diabetes, is not a glucose level, is a response to insulin.
That's brilliant, Luda. I don't think I've ever heard anyone say that in the pain context before, and I think you might be right. Okay, we have time for one more question, and I think this is probably on a lot of people's minds, both concerning this particular story, but just in general. And that question is any of this, anything we've learned from genomic transcriptomic studies, currently clinically useful information? Or if not, will it be, and how soon? What's your prediction as to at what point enough of the variance is explained to provide true clinical direction?
So from what I see today, I don't think that common SNP ever will be the biomarker. Unless, I mean, okay, it can be biomarker for one thing. Okay, sorry, I mean, my mind change all the time, right? So I'm talking about, is it today, is Luda Diachenko. So one thing that can be useful is response to drug. Why? Because we has not been the evolutionary. It was no pressure for us to be chosen one way or the other for drug response. That's why we have example of huge effect size. For example, cytochrome 2d6, and more than 50% of response to cytochrome 2d6 is depending on the number of copy of chromosome cytochrome 2d6. So the other example is what we recently published, the ABC transporter variant explain 80% of our response to morphine or nortriptyline. So this can be huge. Okay, so response to drug, I believe we should continue to look. In other words, it's a gene environment interaction. It can be some other gene environment interaction, but I would doubt. I think we should look in the field of first changeable response to or reduction of. And I think it should be more molecular. It can be small molecules like lipid, can be protein, can be RNA. And I also believe in not one gene or one protein, but in the process. Because what I saw so far is when you start to look at the process, this is much more stable marker, right? Because any specific gene in any particular person going up, going down, not expressed enough. But if you look at the process together, I think this is where we have more stability.
All right, that is a lovely place to end up. And I want to thank you on behalf of myself and the audience today. And I will pass it back to Benjamin.
Thank you, Jeff. Yeah, just to echo you, thank you, Luda, for your fantastic presentation. And those in the audience, again, who contributed to that insightful Q&A session. As a reminder, this webinar will be uploaded to IASP's Pain Education Resource Center for later viewing. Please also, as a friendly reminder, keep an eye out for our September webinar that will be presented by Chief Fu Ma on how the new classification or new way of thinking can translate into clinical practice. Another friendly reminder is that if you are attending World Congress in Toronto, please be sure to stop by and attend the Global Year seminar that's happening on Wednesday. That is the 21st of September from 12 p.m. to 1 p.m. local time in room 604. For more information on this, please visit the Congress website and keep an eye out for our newsletters. And again, thank you all and have a great rest of your evening. Thank you.